So in order to understand the capture of the light energy, we need to define the light first. So light is a form of electromagnetic energy, also called electromagnetic radiation. So like other electromagnetic energy, light travels in rhythmic wave. So light goes up and down in the very particular set wavelengths. So the wavelength is the distance between these crests, one crest to the another. And if it's 700, so the distance is 700 nanometer between these, then it's the red light. If the distance is much smaller, 500, it's blue, 380, it's purple, violet. If it goes much smaller, then we'll stop seeing. We'll not be able to see, but it exists as a ultraviolet light. If it goes much smaller, it goes X-ray and the gamma ray. And if the light energy increases as this the wavelengths shorten. So the short wavelengths, light has more energy compared to the large wavelengths. So in the similar manner, if you go more than 750 nanometer, so if this goes bigger than 750, we'll stop seeing with our naked eye, but it's there. So it's going to be infrared, and if you pass, it's going to become microwave and the radio wave. And as the wavelengths increase, the energy that's carried by this light gets lower and lower. I mentioned that the light is a wave, but it also performs as a particle. We call it photons. So pigments are compounds that absorb light, and there are 4 major pigments in the chlorophyll. We'll just talk about 3: chlorophyll A, chlorophyll B, and carotenoids. So chlorophyll A, before going into the 3, let me tell you, the pigments, the reason why pigments work, has different color, is that they absorb a very specific wavelengths. It doesn't absorb all the lights. It absorbs specific wavelengths and reflect specific wavelengths. So for the case of chlorophyll A, it absorbs around 430, 40 violet bluish, and then it doesn't absorb anything around green, red, yellow, and it absorbs reddish orange. That's chlorophyll A. So another pigment is chlorophyll B, which absorbs more toward the higher wavelengths, so more bluish, and then no absorption around the green and red, yellow, and has absorption around the orange. And then another one is carotenoid. It only absorbs around 450, 60, around the 490, and nothing after that. So if you add all the pigments absorption, it looks like this. That means that around 450 lights are mainly absorbed, but around green, 550, 600, these lights are not absorbed. So these lights are reflected. And then around, the 700, the red are absorbed. That means these 2 peaks are absorbed, but these green or yellow lights are reflected. That's the reason why when we look at the tree, we see green, because that's something they don't want. They reflect, and those lights hit our eyes. These wavelengths, orange and blue, those wavelengths are absorbed by those pigments, so we don't see them. So that's the reason why we see these green in the green.
Thylakoid membrane in chloroplasts contains light-absorbing molecules called pigments. Different pigments absorb light of different wavelengths. One pigment, chlorophyll A, absorbs mainly blue-violet and red light. This molecule participates directly in the light reactions. Another molecule, chlorophyll B, absorbs mainly blue and orange light. We do not see these absorbed colors when we look at a leaf. Instead, we see the green wavelengths that are reflected back from the leaf or transmitted through it.
So we briefly go through the absorption spectrum and action spectrum. So absorption spectra is the value of absorption for particular pigment at any wavelengths, at varied wavelengths. So chlorophyll A absorb lots of lights at this wavelength, this color, but not so much around these, and it absorbs a lot at this color of the light. And each chlorophyll and pigments have particular specific profile. So, and which one works the best for the photosynthesis? So chlorophyll A is a primary pigment. So this is the major pigment that capture the light and then drive the photosynthesis. Chlorophyll B, this one, and the carotenoids, the orange one, those are called accessory pigment. They are not really driving the photosynthesis, but rather they capture the other spectrum that can be supplemented to the chlorophyll A spectrum. So chlorophyll A cannot utilize this wavelengths, but chlorophyll B can absorb, or carotenoids can absorb much larger wavelengths light. So it's complementary. But it can also, so those accessory pigments can also work as the protectant, so photoprotection, because sunlight can also be damaging to the cell when it's too bright. So that's why carotenoids and other accessory pigment can absorb the light and prevent the damage to the cell. So that's the absorption spectrum. Action spectrum is a little bit different. You don't measure the absorption, but you measure the rate of photosynthesis. So this shows that overall which spectrum is used for the photosynthesis, regardless of all those pigments. So this shows that this violet blue light is highly used. So if you shine this light, the photosynthesis can happen, but if you shine the green and yellow light, they do not really do the photosynthesis because they cannot utilize this color of the light. And if you go orange red, the plants start to do the photosynthesis because they can use these wavelengths. So this is the difference between absorption spectra and action spectrum. So this is how the chlorophyll looks like, and it has a porphyrin ring, which is the light absorbing head of the molecule, and it has magnesium atom at the center of this porphyrin ring. And we had chlorophyll A, but the difference between the chlorophyll A and B is just this portion. So this portion is CH3 in the chlorophyll A, CHO in the chlorophyll B. But that can change the absorption spectrum of these molecules. And this chlorophyll has head and also tail. So this tail is the hydrocarbon tail, which is hydrophobic. Why? Because this has to be anchored on the thylakoid membrane of the chloroplasm. So the thylakoid membrane is the hydrophobic, so it has to have hydrocarbon hydrophobic tail, so that this molecule gets anchored on the membrane. And this is the space-filling 3D model of chlorophyll A. So you can see this magnesium inside the ring, and you have the tail, hydrocarbon tail. So what is the basis of energy capture? The chlorophyll has to capture the light energy and convert it into the chemical energy. So when photon, the light energy hits the chlorophyll, this is more the pigment, right? The electron of the chlorophyll in the shell becomes excited. This is the ground state, which is stable and low energy, to the high energy unstable state, which is called excited state. But this is unstable, high energy, and it wants to go down, so that this is the low energy and stable state. It happens soon. But there's a difference in terms of free energy. So low energy, high energy. When this excited electron falls down to the ground state, lower energy, the difference in the free energy is released as heat or photon. And this energy, the difference in the energy, can be harnessed as the chemical energy. So this is how the light energy can be converted to the chemical energy using the chlorophyll molecule. So on the surface of thylakoid membrane, so as you remember, this is the chlorophyll, and you have sacs, the membranous sacs, and you have stacks of them. And one of the membrane, the thylakoid membrane, is magnified here. So you have phospholipid bilayer, and embedded are the light harvesting machines called photosystem. This is the photosystem. It looks like an apple. And the center of the apple, the core of the apple, is the reaction center complex. This is where the things happen. And surrounding is the light harvesting complex. Both of them, this reaction center complex and the light harvesting complex, both of them are protein complex. Embedded are these chlorophylls, or it could be other pigment. So inside this is reaction center complex, there are two, always pair, of chlorophyll A molecule embedded, and these are the special chlorophyll A. And embedded in these light harvesting complex are these multiple pigment molecule, which include chlorophyll A, chlorophyll B, and all other carotenoids. So these are these receive the photon. So you have the light, that light hits one of these pigments, and then excites electron. The energy is transferred until it is transferred to the special pair of chlorophyll A molecule inside the reaction center complex. Once the energy hits here, the excited electron can be transferred to other molecule, not to the other pigment, but the other molecule other than pigment. And in this primary electron receptor, an electron acceptor can receive this excited electron and get reduced. So this is the first step of light reaction. And it happens on this thylakoid membrane, and you see outside the stroma, you have the thylakoid space. And there are two kinds of this photosystem. One is called photosystem 2, that has a complex called P680, the reaction center complex P680. Another one is called photosystem 1. So 2 is the first, and then 1 is the next. and it has a reaction center complex called P700. So these are the two kinds of photosystems. First one is called photosystem 2, and second one is called photosystem 1. And you start with photosystem 2, and then the energy goes through, and it goes to photosystem 1. It's very not intuitive, but the photosystem 1 was discovered first, and photosystem 2 was discovered second. That's why even though the photosystem 2 comes first, it's 2. Right. So the photosystem 2 functions first and is best absorbing light at wavelengths of 680 nanometer. That's why the chlorophyll in the reaction center is called P600. In the photosystem 1, so this is photosystem 2. Photosystem 1, the chlorophyll of the reaction center absorbs the light best at 700 nanometer. So that's why this chlorophyll in this reaction center is called P700. So this P600 and P700 are nearly identical, but they are associated with different proteins, so that's why the absorption light property is slightly different between these chlorophyll in the photosystem 2 and the photosystem 1. So the light capture and the series of electron flow. So you capture the light and the series of electrons. This is the electron flow through the photosystem 2 and 1, and other complex is called linear electron flow. And it happens on the thylakoid membrane. The objective of this linear electron flow is to produce ATP, which is useful for energy transfer, and NADPH, which is useful for capturing the electron. Yes. So making ATP, making NADPH, these are the objectives of the linear electron flow. So linear electron flow can be broken down into series of steps. We have 8 steps. From 1, go through everything to 8 to create NADPH. Right, so let's start. A photon hits pigment. See, you have the photon, it's the light energy. It hits one of these pigment, but this cannot transfer electron. So energy transfer, hop until it hits the P680. This is the one in the electron reaction center. Yes. So this excited electron from P680 is transferred to the primer electron acceptor. Now it's called P680 plus. Plus denotes the activation. So when that happens, now you can split water. You have water coming in, and the enzyme splits. So H2 is split by this enzyme. Electrons are transferred from the hydrogen, so you have hydrogen atom to P680. So that means you have 2 electrons leaving. You have the hydrogen atom will become hydrogen ion, losing 2 electrons, and oxygen is released as a byproduct, waste product. But we animal use oxygen to do the respiration, so that's great for us. So after this step, you have high energy electron. This high energy electron can go through the series of electron transport chain and lose the potential energy. and it can release the energy and drive the ATP. So let's see the detail. So this is the photo system 2, the one that we were talking about. So you have split the water into oxygen and hydrogen atom and electron. Electron is activated here and goes through the electron transport chain, and if it goes through this downhill of energy, it can the energy can be used to transport hydrogen from the stroma to the thylakoid space. So we created hydrogen ions here, and we also imported hydrogen ions here. So inside, it gets very high in the hydrogen ion, and these hydrogen ions want to get out. So there's a force. It creates a force based on this concentration gradient. Now we have ATP synthase, which is like a turbine. So by passing these the hydrogen ion from very high concentration to the low concentration, it can rotate and it can generate energy. So this energy can convert ADP and inorganic phosphate to create high energy ATP. So this ATP now can go into the Kelvin cycle. So we'll finish for the system 2 and the electron transport chain ATP synthesis. Now we have photosystem 1. So the light hits one of those pigments, the energy, the light energy gets transferred, and it hops until it hits the P700. Then the P700 gets energized, activated. Now we call it P700 plus denoting the activation status. Once it gets activated, it can accept the electron from and pass down from the photosystem 2 and the electron transport chain. Even though they went through this electron transport chain and released most of the energy, now it's low energy electron, but it can get transferred to the P700. And the P700 can activate this electron much higher. So now you have highly activated electron that can do lots of work. So excited electron falls down again, right? So those electron transport chain pass down this electron, and the FD, the ferrodoxin, receive this electron transferred to the NADP reductase. The NADP reductase will reduce the NADP positive and hydrogen positive. So this is hydrogen ion. So you have two electrons going into these NADP H plus and H plus to create NADPH. So this is oxidized form, and this is reduced form with two electrons inside. So at the end, this process also reduced the hydrogen ion from the stroma, which is good because you want to create this concentration gradient between the thylakoid space and the stroma to generate this ATP using the ATP synthase. Right, so at the end of the light cycle, what do you get? You get ATP and NADPH. And those two go into the Kelvin cycle. So this entire linear electron flow can be represented as this analogy of a mechanical process. So now first you have photosystem 2. Photon hits the electron. Now the electron has a lot more energy and it can do more work. So the electron goes through the electron transport chain, and through reducing the energy it can generate ATP. Mill, it's like a mill, making the ATP using by rolling the electron down. Now it has lower energy, and go to the photosystem 1. You get the high energy again by getting another photon energy. Now you have high energy electron again, and this can be used to create NADPH. So at the end you create ATP and NADPH using two kind of photo energy input, and through passing this electron up and down. So this is the summary of light reaction and linear electron flow.